Welcome back to Biochemverse by Hammad. In today's lecture, we are going to explore one of the most essential biomolecules, that's proteins. And here's a short introduction about protein. That protein are the true machinery of life, as we discuss in our upcoming slides in few minutes. And they build our muscles, defend against infections, carry oxygen in our blood, and catalyze the biomolecule reactions that make life possible. These all are the totally different reactions and procedures in our body that carry out, but the protein is involved in them. And so protein is involved as the structure of every protein varies accordingly. So we'll discuss what protein are made of, how amino acids link together to form them, and how their structure determine their amazing functions. So let's move forward about the introduction to proteins. As we have discussed previously in our previous two lectures about carbohydrates and lipids, is the same. It's also biomolecule protein. They were also the polymers like oligosaccharide, polysaccharide. Then the lipids have the forms. So the protein is also a macromolecule composed of long chain of smaller units known as amino acids. They contain four major elements. As I'm giving the reference of both carbohydrates and lipids, and that it will be more understandable to you, that you will remember it for a long time. Like in carbohydrate and lipids, they contain three major elements like carbon, hydrogen, and oxygen. Uh, but in proteins, they contain the same three elements, but there is a fourth one also, the nitrogen. But sometimes it's sulfur and phosphorus also. We'll discuss this later in our next lectures of metabolism of amino acids and proteins. So there is a short key box, like if carbohydrates provide energy and lipids store it, proteins are that one that uses it for structure, communication, and catalysis. Here are some core functions of proteins, like enzymatic, structure, and defensive. As we have previously discussed, that it works in many communication, and is also enzymatic effects and defensive. So protein make up approximately 50% or more of the dry weight of most cells. Every cell functions from DNA replication to muscle contraction is either carried out or regulated by protein, because most of the enzymes are protein. And also it gives us structural uh, formation to cells like collagen, and also defensive uh, property of protein, like it's protecting body, for example, like antibodies. So the major element, amino acids, the building blocks that combine together to form a protein. The building blocks of proteins are amino acids. Each amino acid has a central alpha carbon, and which is attached to four different groups. An amino group, and it's a basic property, gives a basic property. And then a carboxylic group that gives the acidic property, and a hydrogen, that's a constant part that never changes. And then the side chain, or that determines the identity, a variable side chain. So the general formula of amino acid is ammonia, then a central carbon, then a constant hydrogen, along with carboxylic group, and then a side chain R, that is variable side chain. The amino groups give basic property, while the carboxylic group gives acidic properties, and hydrogen is constant. And why that's why amino acids are called amphoteric, because they show both properties, basic and acidic as well. So they can act as both acids and bases. There are few examples I have mentioned here: the glycine, alanine, and cysteine. Glycine, simplest amino acid with R chain, is only hydrogen. So if All the constant around central carbon are hydrogen, ammonia, and amino group and carboxyl group. Then the side chain will be only hydrogen that forms glycine. That's the simplest amino acid. So the next one is alanine. It contains the methyl group in its side chain R. Then the cysteine, it contains sulfhydryl group uh, that forms disulfide bond in proteins. So let's move forward. Zwatrin state amino acids as buffer. In aqueous solutions, amino acid exist not as simple molecules but as in aqueous solution, amino acid exists not as simple molecule but as a zwatrin molecule with both positive and negative charges. The amino group accepts a proton and becomes um, positive amino group, and while carboxyl group donates a proton to become negative carboxyl group. So the structures become like that. Amino group accepts the hydrogen released by, and carboxyl group releases hydrogen and becomes negative. So this property allows amino acid to act as buffers, maintaining the pH of biological system, a concept we have discussed previously um, in our water and pH lecture. So think of them as molecular diplomats. uh, Diplomatic property they have. They can also become acidic or basic and maintain the pH of our biological systems. The classifications of amino acids, as amino acids are of 5 different kinds. 20 amino acids, there are 22 amino acids generally, but generally not. Generally they are 20, then other two are also there because the variable side chains they indicate, and also the types varies because of their side chain. So depending on their side chains, amino acid can be grouped as non-polar, polar, acidic, basic, and aromatic. So non-polar amino acids like valine and leucine are hydrophobic. They prefer to hide in the interior of the protein. So and polar amino acids like serine and threonine and cysteine interact with water and form hydrogen bond. Acidic amino acids such as aspartic acid, and also known as aspartate, and glutamic acid, also can be named as glutamate, contain an extra carboxylic group, while basic amino acid like lysine and arginine have an extra amino group. So they gives more acidic property. So they are categorized as acidic because they have extra carboxylic group. That's why they are categorized as acidic, because they are giving more acidic property, and they have extra amino group rather than the amino group attached with the central alpha carbon. So they are positively charged at physiological pH. So the next one is aromatic, like tryptophan and tyrosine contains a benzene ring. They can absorb ultraviolet at 280 nanometer. This property is often used to measure proteins concentration in lab spectrometers. Non-polar side chains amino acids include glycine, which contains side chain only hydrogen, the simplest as we have discussed before, and then the alanine, it contains the methyl group in its side chain, and then the valine, and then leucine, isoleucine, phenylalanine, tryptophan, methionine, and proline. Phenylalanine and tryptophan are also categorized as aromatic, but they are also giving the non-polar property because of their hydrophobic behavior. Then the polar side chains, they are the uncharged polar side chains include 6 amino acids that are serine, threonine, tyrosine, asparagine, glutamine, and cysteine. As tyrosine is also mentioned in the aromatic and also be in the polar uncharged because it has the hydroxyl group at the end of the benzene ring attached to the methyl group with the alpha carbon. And cysteine has also some specific property in the polar side chains. It has the methyl group attached with alpha carbon and also a sulfur hydro group attached with the methyl group. And glutamine has also an amide group and extra amide group at the end of the side chain R. Then the acidic side chains include aspartic acid and glutamic acid, that are also known as aspartate and glutamate. And in the basic side chains, it include histidine, lysine and arginine. So these are the 20 amino acids that are considered majorly. So moving forward, the linkage of bond formation. When two amino acids combine, the carboxylic group of one amino acid react with the amino group of another. This condensation reaction eliminates one molecule of water and forms a new bond known as peptide bond. As I have mentioned here in this, you can see that peptide bond, there is one amino acid. These are the constant side chain, these are the constant, and we have just to take one amino group of one amino acid and carboxylic group of another, and they will combine by donating water molecule as hydroxyl group from carboxylic group and one hydrogen from amino group, and they are making hydrogen and carbon nitrogen bonds, peptide bonds, which will release water and will form a peptide bond between two amino acids, and so on they will move on as much of amino acids should be added in a protein as per their coding. So there was a chemical reaction. This chemical reaction to form a polypeptide chain, the foundation of every protein. In proteins, the polypeptide bond is planar and rigid due to resonance between the carbon and oxygen double bond and carbon nitrogen bonds. The nitrogen carbon and then another carbon attached with the carbon backbone repeats along the chain, as so this repeats so on again and again to the and forms a long chains of the polypeptides of proteins. So moving forward, the peptide bond structure has partial double bond and character due to resonance, and a partial double bond character due to resonance. The lone pair of electron on nitrogen delocalizes towards the carbonyl group. This restricts rotation around the bond, giving the polypeptide a stable and predictable structure. This is one reason proteins fold in specific pattern, not randomly. So that's the point, that every protein have the specific structure, not every protein are similar to each one. So the next one is levels of protein structure hierarchical, uh, primary, secondary, tertiary and quaternary. First of all, protein structure can be understood in four hierarchical levels, as I mentioned here. Primary, so let's discuss about it. The linear sequence of amino acid linked by peptide bonds. For example, the sequence in insulin determine how it will fold and function. And the second one is secondary structure, local folding pattern stabilized by hydrogen bond between backbone atoms. The two main types are alpha helix, where the chain coils like a spring, as in keratin, and beta pleated sheet, where strands align side by side, as in silk fibron. And then the third one is tertiary structure, and the overall 3D folding of single polypeptide chain maintained by hydrogen bond, ionic interactions, hydrophobic forces and disulfide bridges between cysteine residues. Then the fourth one is, and the last one, quaternary structure, association of two or more polypeptide chains into a functional protein. Hemoglobin is the perfect example. It has four subunits, two alpha and two beta chains. So these were the hierarchical levels of protein structure: primary, secondary, tertiary, and quaternary. Moving forward, denaturation of protein. It's a short introduction about denaturation of protein, and we will discuss it elaborately in the degradation of amino acids, and also in the metabolism of proteins, metabolism of amino acids. When proteins lose their structure, they also lose their function, a process known as degeneration. Degeneration can be done by many processes, as in the egg. Take an example of egg. It denatures proteins when we heat it. So denaturation can be caused by heat, strong acids or bases, alcohol, or heavy metals. A familiar example is when you cook an egg, the clear egg white turns opaque white because the protein albumin unfolds and aggregates. In our body, high fever or abnormal pH can similarly denature enzymes and disrupts our metabolism, as we have discussed in our lectures of water and pH about it, that how instability in pH can affect our metabolism in our body, and causes many kind of the diseases. So the biological roles of protein. Protein perform an incredible range of biological function. Structure proteins like collagen give strength to bones and tissues. Enzymes like amylase and catalase speed up vital biochemical reactions. And for in the transport in transporting compounds, elements, and many other things, like hemoglobin transport oxygen in our blood, and while antibodies defend against pathogens, can be the property of the protein in a defensive way. And hormonal protein like insulin regulate metabolism. That the regulatory role of protein. And ferritin store iron safely inside cells. That's the storage property. So the main five or six properties that protein perform: structural, enzymatic, transport, defensive, regulatory, and storage. So to summarize, proteins are polymer of amino acids. Amino acids join via peptide bonds formed through condensation reactions. Proteins have four levels of structure that determine their function, and denaturation disrupts structure and leads to loss of activity. Proteins are vital for almost every process in living organism, from catalysis to structure to defense and transport, as we have discussed here. So there is also, I'll give you some example, that your body contain over 1 lakh different proteins, each with a unique sequence. There is a code that construct the amino acid polypeptides. So every protein have the unique structures. So there are 1 lakh different proteins. The longest known protein is titin, has more than 38,000 amino acids, and enzymes can accelerate reaction by factors of 10 raised to power 17 times more. That's like speeding up rusting iron into a blink. So hair, nails, and feathers are mostly made of keratin, a tough protein rich in cysteine. So every time you crushing your nails or combing your hairs, so take a notice that these are the proteins. So that's all for today's lecture on protein, amino acid, peptide bonds, and level of structure. In our next lecture, we'll explore nucleic acid, RNA, and DNA, and we'll learn how protein makes life reactions, happen at lightning speed. So don't forget to like, share, and subscribe to Biochemverse by Hammad at YouTube, and at Facebook its name Hammad Bashir Khan. And for more engaging biochemistry content, stay curious, stay scientific, and see you in the next lecture. Don't forget to share our content with your fellows and with your siblings, offering their studies and their efforts in the field of biochemistry, molecular biology, or in the field of medicine, medical. So that's it. Thank you so much. See you in the next lecture.